This production has been brought to you by the Fresno Mycology Society. Welcome to the first in our Mycology 200 core series, which will focus on phylogeny, taxonomy, ecology, and the morphology of fungi. We will start from a broad fundamentals level and focus in from there. If any of this information is redundant to you, I've included timestamps for you to easily navigate the material. Our goal is for everyone and anyone to be able to comprehend the preceding material. To do this, the fundamentals must first be discussed. I'll do my best to define any jargon or scientific terminology as it comes up, but if you ever need further clarification for the definitions of words used in this series, I've included a link of a brief glossary of terms down in the description. So right up top, let's define some general terms and concepts. Starting with the fields we'll be discussing, taxonomy is the science of classification which places organisms into a hierarchical system known as Linnaean taxonomy. This system, created by Carl Linnaeus in 1753, moves from large to smaller groupings of organisms until you eventually arrive at the species. In this way, the further down the chain you go, the more similar things begin to look to one another. Let's look at these groups with an example we're all familiar with, the portobello mushroom, Agaricus bisporus. Starting at the broadest group, we find portobellos, puppies, plants, and insects in the domain Eukarya. All very different, but united by the fact that they have cells with nuclei in membrane-bound organelles. The next grouping, Kingdom Fungi, obviously contains all the funguses, or fungi, if you want to be proper. This broad grouping includes yeast, mushrooms, molds, chytrids, and others. Next, the phylum level grouping, Basidiomycota, contains the mushrooms, puffballs, polypores, boletes, jelly fungi, and a few others. Then, fast forwarding through class Agaricomycetes, order Agaricales, family Agaricaceae, to the genus Agaricus, where things finally start to look a little more similar to one another. All generally have caps held up by stems, gills, and produce brown spores. No puppies here, sorry. Finally, we're arriving at the last and the most specific taxonomic level, the species epithet, of which portobellos are bisporous. But an important thing to know is that you would never refer to portobellos simply as bisporous, as species epithets are reused across the tree of life. Instead, we want to also include the genus as well, in what is known as binomial nomenclature, which allows us to triangulate the organism. Therefore, the proper scientific name for portobellos is Agaricus bisporus. Okay, now we can move on. The next term, phylogeny, is simply the science of relatedness, which is semantically quite similar to taxonomy, but where taxonomy is concerned with the names of the organisms on the tree of life, phylogeny is concerned more with the pattern of branching that arrives at each name. How is portobello related to other species of Agaricus? What was the most recent common ancestor? How are the traits that we see in portobellos derived from their less delicious ancestors? These are the questions phylogeny is concerned with. Molecular systematics is the discipline of phylogenetics which examines the genetic molecules, or the DNA, of organisms, primarily through genetic sequencing to elucidate their relatedness to other organisms. And lastly, morphology is the field of science that describes the forms and features of organisms. If you want to know more about the morphological features of fungi, reference our Mycology 100 series. So now we can discuss the topic of this video, the Kingdom Fungi. With a myriad of shapes, sizes, and ecological niches, what is it that defines the kingdom of fungi? Traditionally, fungi were defined as any eukaryotic, achlorophyllous, spore-producing organism with a filamentous somatic structure. However, if we're to stick to this categorization scheme, the kingdom fungi would also include Oomycota, aka the water molds, and slime molds, which we now know belong to the kingdom Chromista and the unicont phylum Amoebozoa, respectively, but are often called fungus-like organisms. Therefore, we must make a far more rigorous definition for the true fungi, sometimes referred to as the Eumycota. To continue forward with the previous definition, True fungi also obtain energy by non-phagotrophic absorptive nutrition, the process of secreting enzymes onto the substrata and absorbing the smaller digested molecules through the cell membrane. This additional criteria eliminates the slime molds from our definition, as they make use of phagocytosis to feed. True fungi also have cell walls made of chitin, which differentiates them from the water molds, which have cell walls composed of cellulose. True fungi also never have an amoeboid phase in their life cycle, exhibit asexual means of reproduction, and have short-lived diploid phases. The molecular systematics of the Eumycota shows us that they are phylogenetically sister to Animalia, and are united under the superkingdom Opisthokonta, which translates to posterior tail. In fact, similarly to the sperm cell seen in Kingdom Animalia, the Chytridiomycotan fungi still maintain the ancestral trait of having posteriorly flagellated zoospores, a trait which has been lost in all the other fungal phyla. So with all these criteria in place, we've arrived at our target kingdom, the fungi. This group is strongly supported by molecular phylogenetics to be a monophyletic group, meaning that they are all descended from a common ancestor. In contrast, the traditional definition of fungi we mentioned earlier would be an example of a polyphyletic group, or a grouping of organisms that does not share a common ancestor with all its descendants. This is bad. If you submit a paper of a molecular phylogeny of organisms that looks like this, you will lose your grant, because it's considered an erroneous or artificial grouping that does not help to convey the actual ancestry of the organisms in question. Currently, it is thought that fungi may be the most numerous eukaryotic organisms on the planet, perhaps only second to animals, the bulk of which are arthropods. This is an estimate, of course, as we've only formally described around 120,000 species, but given the density of species diversity in well-sampled areas, we can infer that 1 to 5 million species of fungi exist on Earth. This means that for every one species that we know, there's around 10 to 40 that are not described. This estimate expands every time we find species diversity of fungi in previously unimagined places, such as the depths of the seafloor around the world, in the microbiome of ourselves and other animals, and inside the tissues of every plant ever sampled. At the kingdom level we're focusing on in this video, the morphological diversity is massive, from single-celled yeast to the largest organisms on the globe by area. The diversity is truly vast. Some taxa are highly cryptic and never produce macroscopic fruiting structures, while other species, such as Phelinus ellipsoideus, form massive fruiting structures, the largest of which weighed in at over 1,000 pounds. But, aside from the single-celled exceptions, almost all fungi are united by the filamentous body organography. These fine threads are called hyphae, a mass of which constitutes the mycelium. Hyphae have cell walls composed of chitin, a resilient long-chain polysaccharide made from N-acetylglucosamine, a derivative of glucose. Fungal mycelium can be so densely branched that a single cubic centimeter of rich soil can contain over 1 kilometer of hyphae laid end to end. The ultra-thin nature of fungal hyphae is the key to their success, as this allows them to maximize the surface area of contact with the substrata they are actively digesting. One could argue that the major evolutionary divergence between Animalia and fungi was where animals evolved internal digestion, generally taking advantage of the digestive power of a diverse microbial flora. Fungi, instead, opted to externalize their digestion. As such, the mycelial network can be thought of as a completely externalized digestive system, secreting digestive enzymes onto a food source, which breaks down the chemical bonds of complex polymers into their constituent monomers, at which point they are able to be absorbed into the mycelial cytoplasm through active and active-passive or facilitated diffusion. There are two primary types of hyphae, septate and aseptate, or cenocytic hyphae. Cenocytic hyphae, as seen in the Zygomycota and the Chytridiomycota, are tubular filaments that lack septa, or divisions. You can essentially think of them as massive, multinucleate, single-celled organisms that are hyper-thin and highly branched. In contrast to the hyphae of, say, a mushroom, which are septate, the cytoplasm of the mycelium is still continuous throughout the whole thing, as it flows between septa through central septal pores, but the nuclei and organelles are confined to their individual septated cells. In their natural environment, fungi occupy a huge variety of ecological niches. Most are saprotrophic, meaning they feed on dead organisms. Some are parasitic, and many more are mutualists, and live in a community of give and take with other organisms. If all fungi were to disappear off the globe in a moment, planet-wide ecological collapse would rapidly occur. Many species of fungi are parasitic and lead to the premature death of many plant species. While our first response toward parasitism is often very negative, and in the case of non-native invasive parasites, they almost always are, however, in their natural environment, parasites are essential in maintaining a healthy ecosystem by increasing habitat turnover and preventing any one particular species from dominating over the rest, thus encouraging diversity. Environments composed of fewer species are almost always less resilient to disturbance than diverse ecosystems, as a host of species provides niche redundancies and creates buffer regions that prevent a pathogen from dominating. Another role fungi play is that of the mutualist. Though there are many, the most ubiquitous example is that of mycorrhizal fungi, which are species of fungi that symbiotically attach to the roots of 80% of all known plant species. In this partnership, the plant donates carbohydrates in exchange for water and mineral nutrients to and from the fungal symbiont. Many species of plants cannot subsist in their environment without these mutualists. Another fascinating example of fungal mutualism is the fungal endophyte, a universe of relationships that are understudied and still poorly understood. What we do know is that every species of plant sampled to date contains endophytes of some sort, that is, organisms that live within plant tissues that do not cause disease. These endophytic fungi, as well as many bacterium, can be thought of as the plant equivalent of a microbiome, and impart an array of host benefits, such as disease resistance, drought tolerance, heat resistance, and produce constellations of secondary metabolites, which can aid in anti-herbivory, chemical signaling, and increases the host's ability to uptake nutrients. To reflect on the ecological relationships of fungi is to realize that the individual, as we often see it, is merely a macrocosmic expression of a vast plurality of microcosmic relationships. Though it's the human inclination to see species and their environments as separate individuals, next time you look at a tree, reflect on the universe of microbes both inside and outside of that tree, without which it could not exist. It's good for you, I promise. The last and arguably most important ecological function we'll discuss, though there are certainly countless more, is the role that fungi play in the carbon cycle. All life on Earth, as we understand it, is carbon-based. Elemental carbon composes as much as 50% of the dry biomass on Earth. Carbon-based molecules, or hydrocarbons, are utilized in every aspect of life. The basic unit of energy used by the cells of all organisms is glucose, a simple carbon-based sugar molecule. And the most abundant molecule on the planet is cellulose, which, along with lignin, is the main structural molecule of plant tissues. However, though cellulose is essentially just a string of glucose molecules, few organisms can sufficiently digest the strong beta-1,4 linkages of cellulose. If you were to try and eat wood, for example, you'd get about as much out of it as you would eating a plastic bag. And for the exact same reason. Humans, and most animals in general, do not possess the suite of enzymes necessary to break the powerful beta linkages of cellulose molecules. Therefore, it is vitally important that something releases the carbon bound within plant materials and allow it to continue cycling. Enter the fungal saprotroph. As enzyme-producing powerhouses, fungi, and to a lesser extent prokaryotes, are able to melt the strong hydrocarbon bonds of cellulose and lignin and free the carbon back into the soil and the atmosphere in its bioavailable form. Without the fungal saprotroph, soil production in the terrestrial ecosystems would be nearly non-existent. And as terrestrial ecosystems are literally born from healthy soils, complex ecologies likewise would not exist. The nuances of fungi in the carbon cycle, and more specifically soil production, is vast, and deserves its own dedicated video, but we'll leave it there for now. I hope you found this video enjoyable and instructive. The next video will descend taxonomically from Kingdom Fungi to the phylum level. Preceding videos will likewise work their way down the taxonomic hierarchy. I've wanted to create the Mycology 200 course series for at least 3 years now, but have not had the proper tools to produce them in a manner that I'd be satisfied with. In that time, I've invested much of my own money towards purchasing the software and hardware necessary to pull this thing off, and much of my time learning how to use them. As this channel has grown, some of you have helped with these expenses by becoming patrons on Patreon. If this is something you would like to do, you would have my genuine appreciation, and also probably get a couple cool mycological treats in the mail from time to time too. To do so, you can find us on Patreon at Fresno Mycology Society. You can also find us on social media on Instagram, Facebook, and Flickr. If you're in the Central Valley area and would like to be notified of any mushroom hunts, classes, or events that we hold, you can subscribe on our website at fresnomycology.org. All these links are in the description below. Thank you so much for being here. Until next time, happy hunting.